It's really an honor to have Dr. Markert here. And Jim, the other thing is we'd like to record this if that's okay. I
Perfect.
meant to, I was meant to had to
No
remind me
worries.
to say that, but it's really a delight and pleasure and honor to have Dr. Markert here. He has been in Boston a couple of times in his career. Actually, went to Harvard College, and then went on to Columbia for medical school and University of Michigan for neurosurgery residency, and then returned to Boston at the end of his residency for a fellowship in molecular biology, molecular tumor work with Bob Martuzza at the Mass General, and then set up shop at University of Alabama, where he's been incredibly successful in terms of helping to build the program. He also went and did one final fellowship in molecular work, but since returning to Alabama, has been very successful and able to build into a department. What was it, 2013, 14 became a full department.
Yeah, that's right.
And under his leadership, it's just really completely grown and huge part of the institution. But Jim's work has been in tumor molecular biology and viral vectors, and probably one of the one of the thought leaders in the country on this. And so he's going to give us some insight into his thoughts about neuro-oncology, hopefully some viral vector work and molecular genetics, molecular biology. So Jim, very great, and great of you to do this, and a pleasure to have you.
Well, thank you very much, Chris, and thank you for your kind introduction. It's really fun for those of you who are residents and early on in your career to go through the world of neurosurgery, because Chris and I first met each other back when I was actually doing my fellowship in Bob Martuzza's lab, and Chris was just transitioning. I think you were just finishing chief residency and moving into a faculty position at that time. And it's just really a pleasure to get to know people through their careers and see them progress. And Chris has certainly been a good friend, and it's nice to see what you built there, Chris, too, as well. So congratulations. I'm going to tell you a little bit this morning about some of the work we've done and others. And I'm trying to share my screen here if it'll let me. Now we're going to have that moment where I ask, do you see my screen?
It did it. Yep, it did it. It's great.
Okay, and I'm gonna go into presentation mode. So let's see if it. Are you seeing one slide or two slides?
We do see a little slide on the side, but we see the
Okay,
main slide in the middle.
let me just. Now you should just see one. Is that right?
That is correct. That's great, Jim.
Okay, great. Okay, good. All right, so I'm going to tell you about our work with oncolytic HSV for brain tumors here as kind of a model. And I hope that for some of the residents and fellows and others who are earlier on in their careers, but anybody really, I hope to get you excited about the idea of being a neurosurgeon scientist, because all of us, no matter what we do as our subspecialty interest, private practice, academic, what have you, we all have the best laboratory in the world in terms of what we work with the human nervous system. And all of our patients are, in fact, I should say our practices are great examples of ongoing clinical trials. And we want to make sure that we are studying our patients and our outcomes and learning from them as our practices advance. So I want to encourage all of you, no matter what your particular interest or stick or whatever is, to remember that. And I'm sure you're hearing that there as well. So I'm going to tell you about this oncolytic viral stuff. And I do need to make sure that I show you I have disclosures. UAB required that as we did this stuff, we patent it, obviously. And then when people become interested in it, you get this stuff licensed. So I'm very open about that aspect of things. So we are going to talk to you about the outline of our presentation here today, first of all. And this group, all understands malignant glioma, so that's not anything new. We'll tell you about our initial clinical experience with the viruses and what clinical trials we've got going on, where I think the field is going, and what we have planned for the future as well. So this is the patient that all of us have had in our practices, where you've got somebody who comes in, tricky tumor here in the Wernicke's area. You can map it, take it out, but we all know that there's, residual tumor at a microscopic level present. Patient goes through their chemoradiation and comes back to you with a recurrence like this patient did. And, now world of Dr. Google, patients all come in. I've done some reading on the internet, this one says, and I know a lot more than my oncologist does now. I think I want to be on an immunotherapy clinical trial before it's too late for that. So, so I'm sure you're hearing about all this in your practices as well. And just to kind of give you a setting for oncolytic viral therapy and how it works. So you can use a DNA or an RNA virus, right? Viruses have different flavors, and some actually package DNA, some package RNA, but there are different kinds. And it binds tumor cell, gets into the tumor, replicates its nucleic acids, whether they're RNA or DNA, and then RNA is translated, makes the new viral proteins, and the viral capsids are actually assembled. They package the nucleic acids, lyses the cell, it kills it. In oncolytic viral therapy, it kills the cell. In some of the retroviral therapies that you've heard about, like TOCA 511, the virus itself doesn't kill the cell. So that's dependent upon the suicide gene and drug that's administered. But for strictly speaking, for oncolytic viruses, the viral lysis will kill the cell. And so that's important for a couple of reasons. One, obviously, there's some direct tumoricidal effects. But two, it gives the immune system an opportunity to kind of recognize that there's something bad going on here. And this is just kind of a model of HSV in particular that's a little bit more specific than what I just showed you. But again, this column on the right is what I want to draw your attention to. It debulks the tumor directly by killing the tumor cells. But then suddenly there's all these antigens, like what we call a tumor debris field present. And tumors, as you know, are very stealthy. It's kind of, makes you kind of wonder almost if they're have a little bit of their own life form to some degree. They actually, have constructed sort of an immune system avoidance that will allow them to remain below the level of detection by the immune system by producing IL-10, TNF beta, and so forth. And as a result, they're able to stay outside of immune surveillance. But in fact, when you actually add this viral infection to the mix, you can, as it says in number 3 here, recruit innate and adaptive cellular response to a tumor that otherwise previously was before the below the immune surveillance system. And the fact, despite the fact that IL-10 and TGF-beta and so forth are present, you can actually generate enough interferon gamma and other kinds of signals to allow for an immune response. Excuse me. Sorry, and generate some inflammation. And I think, and here you can see kind of, this is what happens with HSV. If any of you are a particular virologist, the STING pathway is something you're going to be hearing more and more about in the immune system, CGAS STING, and that results in this interferon production here. And then we see some interferon specific genes that are stimulated, cytokines, et cetera, that help this immune response. And what I really want to get across here is, I don't know whether HSV is going to be the solution or not, but we have made incredible progress in the field in that we have taken tumors that heretofore were completely below immune surveillance, and now we've brought them into the area where into the arena where the immune system can actually see them. And when we actually go back and resect some of these tumors that are progressing or pseudo-progressing, we can see a complete change in the immune milieu. And I think this is going to be the basis for us figuring out as a neuro-oncologic community how to combat these tumors. So it's a very exciting time. So let's back up a little bit though and look at oncolytic viruses. There's a lot of different kinds, very many different flavors. The ones that you're going to hear about the most in regards to glioma are listed over here on the left. Probably the most studies have been with HSV1 mutants. We started with G207. I'll talk a little bit about that. While we were studying G207, there was a group in Britain that was studying a similar virus called 1716. We were actually way ahead of them, just as this kind of a side tale here. And we were, but suddenly they ended up catching up with us, and we were trying to figure out how they caught up with us. We ended up publishing our first clinical trial in the same issue of gene therapy that they did. And it turned out that while in the United States, we had to go ahead and do all this primate work before we could go into people with our drug, our new drug, the virus. In Britain, you had to go in people before you could go in primates because of the animal rights people there. So, so kind of an interesting twist on things. So at any rate, but both studies showed promise, and that was exciting. And then we went on, and in our work, we've developed this virus called M032. Across town from you, Nino Kyoko's been working on a Q-Nestin virus. And then we also have another virus here, which I'm going to talk to you a little bit about later, called C134. And then adenoviruses are, another huge entry into this field. We initially did work with the group on Onyx 015, and then the MD Anderson group developed Delta 24 RGD. This is the virus that adenovirus that you've heard about that's currently in clinical studies as well. The Mayo people worked on measles virus. Reovirus was another virus that we studied here at UAB. It was also studied in Canada, and it's been looked at in a number of different tumors. The Duke folks, of course, have made polio very well known in the field. And there are others, and some of these are listed here, you can see. What's been remarkable for me to witness, over the course of my career, this didn't exist, right? I mean, there was some early studies over the course of the 20th century about viral infections in a woman with cervical cancer, for example, who had a complete remission of her disease. But what was new when I had the good fortune, I should say, to work in Dr. Martuse's lab was the idea of using a genetically engineered virus. And there was another Harvard guy, a guy named Don Cohen, who actually genetically engineered HSV with the idea that he could maybe make a vaccine against HSV1, and that was an important thing. There was a cover of Time magazine years ago that said, herpes, it won't kill you, but you won't kill it either. So Don was working on a vaccine for that. And then Martuse's group had the idea maybe we could in fact use this for viruses because it was not for tumors because it was not neurovirulent. And so we got interested in that at that time. So what's good about HSV, if you're looking at all these different viruses, well, it's got a large DNA genome, and we know all about how the virus replicates. There's this kind of a sequential viral replication. It turns out that can be helpful in using as a gene therapy tool. So there's up to 30 kilobases of the genome can actually be taken out because they're not really essential for viral replication. They have specific duties or tasks to allow the virus to be propagated latent and so forth, but we don't need that. We don't actually want the virus to go latent for our purposes. So we can remove that portion of the viral genome, and then we can put other things in to make the virus kind of do our bidding, if you will. And then this is obviously the key point that I was alluding to earlier with Cohen's work, that you can take out neurovirulence genes, but still have a virus that kills tumor cells. And so as a result of that work that initially I did in Bob's lab, another Japanese neurosurgeon came in. I went back and finished my residency at Michigan, and this work continued, and they developed this sort of extra safe version of the virus that I'd been working on that had another mutation in it, and they called it G207. And so Bob and I, by that time, I was a young attending here at UAB, and Bob and I designed this phase 1 study looking at the virus in patients that had recurrent glioma. And we had this extra safe virus because at that time we were concerned we didn't want to set the whole field of viral therapy back by injecting patients with this virus and then having them die of encephalitis. So we made it extra safe so that there wouldn't be headlines about that kind of nonsense. And we proved it. We had patients that came in, recurrent tumors, didn't have other options. Some of these tumors looked pretty terrible. They were hollow hemispheric kinds of things. But we injected them with the virus directly in the operating room stereotactically. And then we started out at 1 million PFU. That just means particle forming unit or active viral particle, basically. And then we were able to dose escalate all the way up to 3 times 10 to the 9th plaque forming unit. So we went from a small dose to a relatively high dose. and the patients didn't seem to have any issues from it. So that was huge. And then we had some patients who did really well. And that, as you know, those of you who are clinical trialists, there seem to be a group of patients that do better than expected in many clinical trials. And so you kind of have to filter that out and figure out, well, is there any real signal here or not? But we had a patient, for example, with a recurrent tumor who lived 8 years after her viral injection. We had another patient who had a recurrent tumor. And of course, we biopsied all these tumors at the time we treated them to make sure we really had recurrent tumor. And this patient died of an unrelated stroke a year and a half, 2 years later, and had an autopsy. They didn't find any tumor left. So it was enough signal that it was exciting. We went on at UAB at that time and did what we call phase 1B study. And this was just to try to learn a little bit more about some specific questions that we had. And one of the things we wanted to learn is, one, can we inject the virus twice without causing some kind of overly robust immune response? Two, can we inject the virus directly into tumor infiltrated brain? Because that's the trick with GBM, right? I mean, we as surgeons can all go in and resect the portion of the tumor that's non-infiltrative. So that's, if all the virus did was kill that portion of the tumor, so what? But we wanted to see if it was safe to actually inject into the tumor infiltrated brain around the tumor. And then third, as a scientific note, we wanted to be able to take tumor and see what was happening in the tumor after we injected it. So the study design was pretty straightforward. I took the patients to the operating room, stuck a catheter in them, injected the virus through the catheter, and then came back either 2 days later or 5 days later, took the tumor out like en bloc with the catheter in place, and then injected the surrounding tumor infiltrated brain with additional virus. So the patients got that second dose that I was talking about. And then we took the tumor to the lab and we did some studies that I'll talk about some more later. And we found out, in fact, that this was also a safe approach. And we did have some patients in this study that did well too. And then finally, this third study I mentioned here. So these guys at the bottom, I haven't been talking about them and I should have, but in this last one, bottom right corner, this guy, Ralph Wexelbaum, was a real genius that I've worked with at University of Chicago, radiation oncologist there. And he figured out that if you gave radiation very shortly after giving the virus, you had a more robust degree of viral replication and a better response. And so we designed this study, and I'll talk about that, where we gave both virus and then radiation. And this is that study, and this is the ancient gamma knife that we're using back at the time of that study. And initially, what I'd hoped to do was to actually give a treatment of the virus and then come back and do radiosurgery the day afterwards. Because, as you know, RTOG looked at radiosurgery in GBM, and they found out that it really didn't make a difference in overall survival, but there were certain groups of patients that seemed to have benefit. And the problem was it was difficult to tease out who you give it to and who you don't. And so it really hasn't become a mainstay of glioma treatment. But I wondered, well, maybe there would be enough of a signal if there was something happened from the virus and something happened from the radiation together that we could actually show a real response. So, well, unfortunately, what I was able to do was just do the part one of the study, which was to give the virus with 5-gray. And then the company that we were working with ran out of funding. And so as a result, we never got to do the stereotactic radiosurgery part. So I always put this picture in to remind me of what could have been, so to speak. So, but we did get 9 patients in, and the patients did well. They didn't have any issues with safety and tolerating both the radiation and the virus together. And we had some nice responses here, and it seemed like a good approach. And here's just some of those patients from that study. And you can see, sometimes 3 months, sometimes earlier. This patient, for example, looks like I resected this patient, but I didn't. This was all just the virus and the immune response. Here's another patient, same kind of deal. Here's a guy who had a malignant glioma right in his motor strip. And a couple months later, 3 months later, you can see you can't even see any tumor, and he was off steroids. So some impressive findings. So when we did this, we actually took advantage in that phase 1B, that middle study that I mentioned with the 6 patients in it, of trying to find out what we could learn more about what was happening when we gave the virus. And so, so there were some interesting findings that we found. And this is what I think, is perhaps the most important part of all these studies is that we can induce an immune response in the setting of very immunosuppressive tumor environment. And I think you all know, right, from dealing with these patients, that they're all, when you have a recurrent malignant glioma, you're immunosuppressed, right? The tumor with the TGF beta and the IL-10 and the prostaglandins makes the environment immunosuppressive. The patients have had steroids. The patients have had chemotherapy. They've had radiation. So this is a very immunosuppressed population. And as part of this study, we actually did delayed type hypersensitivity testing. So the kind of thing that you get when you get your PPD checked for TB, right? So we injected common antigens into these people. We had a panel of about 10, things like Candida, mumps, and things that we've all been exposed to either directly through the disease or through vaccinations or environmental exposures. And of these 6 patients, none of them had a delayed type hypersensitivity response to any of the antigens. So, I mean, that was interesting in and of itself to find out just how systemically immunosuppressed these patients are in terms of T cell response and energy. So despite all that, though, in the actual immune environment, you can see this here. This is staining for patients that had their tumor biopsied. So before treatment, these are recurrent tumors. And then 2 to 5 days later, we went back and looked at the tumor after treatment, okay? And here you can see we were staining for CD3. So that's a lymphocyte marker. It gets, T cells in particular, but it doesn't really subdivide into what type. And you can see here, before treatment, there was barely any lymphocytic presence here. And then after treatment, they were variable. Some were a little bit less, but some were more, depending upon where, in fact, we got the biopsy specimens from. So that was kind of cool to see that. And then we went and specifically looked at CD8 cells and had some of the same findings, not so much present by immunohistochemistry beforehand, but afterwards, we saw a lot more. And then when we actually did like a DNA microarray analysis back then, this was before the days of next-gen sequencing and so forth, we found out that, yeah, the genes changed, the gene expression profile changed. And it was different if you looked at the patients before, patients who didn't respond well. So there was clearly a group of patients that, the tumor just motored on after treatment versus the ones that were long-term survivors. And we could see, in fact, that there was 2 different kinds of genes that were upregulated, to some degree apoptotic genes, but really immune response genes were the relevant finding. So, we held on to our specimens, and one of the issues that we had is that, how do you do pre and post when all you've got is a stereotactic biopsy from pre, right? And you can't really get a lot of remnant tissue from that. You can't, in terms of research, I've never felt like it was ethical to say, I'm going to take a whole bunch of stereotactic biopsies and risk a hemorrhage. So what we did was we took what we felt like we needed to, make sure that we were going to have a diagnosis, but we didn't take, 10 or 12 stereotactic biopsies from these patients, just, 3, 4, something like that. But that wasn't enough tissue to do much with. Well, lo and behold, technology advanced over the time period that we held on to these. And suddenly next-gen sequencing became available, nanostring and some other more sophisticated approaches. So this is a group that I work with, Kevin Cassidy and Katie Miller at Nationwide Children's Hospital. And they actually helped us go back and look at patients' tumors. So from those stereotactic biopsies pre-treatment and compare them to specimens post-treatment. And they use this technique called principal component analysis, where you can actually it's a vector-based statistical system for trying to simplify the huge amounts of data that come from analyzing 500 different genes that have been upregulated, downregulated as a result, you hope, of your treatment, right? So we did this with our treatment, and we found out some important things. So if you look at this group, here's the patients that were enrolled in the trial, okay? Now, we weren't able to get DNA or RNA from all of them because it was so long, years and years after the study, that some of the specimens were degraded. But we were able to get 3 for this particular study from pre, and all 6 from post-treatment. And when you look at principal component analysis, you're trying to say, well, which groups are most alike? And what we found out was that the most common changes in gene expression, right? So if you look at how the gene expression trained pre and post treatment, It turned out that was much more important than what kind of tumor it was. So, just when you look at all comers, you think of, well, mesenchymal tumors are all going to share similar gene expression profiles, and classical tumors will express similar genetic profiles, et cetera. But when we looked at this, it was much more, the group of untreated patients looked much more similar in terms of their RNA expression than they did from all the post-treatment. There was a big variability. Now, there's obviously variability between gene expression profiles here post-treatment because some of these tumors are different. And you can see that the mesenchymal tumors tended to group with similar genetic profiles, and the ones that were not mesenchymal tended to group. But still, the similarities were much more between pre-treatment versus post-treatment. And I think that was an important finding. So there's another machine learning technique called unsupervised hierarchical clustering. And this is a way of where the software package just basically looks at different gene expression profiles and starts grouping people. And you can see this dendrogram up here, how the grouping occurs. What's alike, what's different? What's alike, what's different? And so it goes through the sequential paradigm of grouping genetic profiles into more alike and more distinct. And when you did that for our patients, what you found out again was that if you look at pre-treatment in the blue over here, okay, and post-treatment in the yellow, the pre-treatment all by this method also had much more similarities than the post-treatments in terms of gene expression. And again, if you look at mesenchymal versus proneural, you can see that a lot of the mesenchymals did group together, but it wasn't as good a means of grouping similar gene expression profiles as pre and post. So it's interesting findings, and you can see obviously this is a heat map of different gene expression, the 500 different genes that we looked at that had changed the most between pre and post treatment. So kind of an interesting, again, confirmation that maybe we were onto the right track. So we wanted to look at another technique, though, just to make sure that we weren't missing something. So for those of you who are not molecular biologists, RNA-seq and this next-gen sequencing is really a strong tool, but it has a downside, and that's all this, PCR amplification that's necessary. And so you might take something that's not very relevant and amplify it all up and make it look like it's really relevant. Okay, so nanostring is another tool that just looks at single molecules. So the amplification process is not present. The advantage of RNA-seq is that if you have a really low signal, you can catch it because of that amplification process, but you may make more of it than it really is. So it's nice to have both techniques. They kind of balance each other out, if you will. They have non-amplified and amplified. So we looked at nanostring here, and I don't want to belabor this too much, but again, pre here is in dark green and post is in light green. And sure enough, when they did this hierarchical clustering approach, this machine learning approach to try to group these by dendrogram into similar and different gene expressions, you can again see that the post-treatments all grouped together by nanostrings and the pre-treatment all grouped together as well. So kind of a nice confirmation of the amplification approach from RNA-seq. So using nanostring, it's kind of cool. You can actually go and predict by looking at the gene expression profiles, you can predict what kind of immune cells are present in a milieu, in your tumor microenvironment. So, and so this is an important thing. So you can actually go through and look by our patient, and here you can see the patients are listed as well as whether they're pre or post treatment again. And what you can see here, and then also the non-responders, the patients who lived less than 6 months, all sort of grouped together in their post-treatment with one pre-treatment having a similar immune profile. But the responders tended to group together in either one or two different approaches to the immune cells that were present. And again, if you look at the post-treatment, they all had this kind of same increase in expression of certain different cell types. And you can look over here, and I'm going to go into them in more detail in a future slide, but they're kind of what you might expect. CD8 cells are up. Interesting, B cells were up, dendritic cells were up, in the patients after treatment compared to pre-treatment. And when we look at those in a little bit more isolated fashion, because that's kind of an overwhelming way of looking at it, you can see that here's the pre-treatment and the post-treatment, and there was more T cell expression or profiles that were consistent with T cell expression in the post-treatment. If you look at exhausted CD8 cells, interestingly, those were not different between the pre and post-treatment. So that's something we need to think about. What does that mean? Does that mean that we need to, for example, add a checkpoint inhibitor to our treatment? That's something we're thinking about doing. So actually going to do, I should say. And then here's dendritic cells. So obviously these are necessary for antigen presentation and developing a Th1 type response. And sure enough, they're different, significantly different post-treatment. NK cells are also significantly different post-treatment compared to pre-treatment. And again, I'll just show a few more. TILs are really the big difference. And I want to talk to you about this just for a minute, because I think we're all aware of the fact that if you look at particularly melanoma and maybe non-small cell lung cancer, these checkpoint inhibitors have really changed the face of outcomes in therapy, right? We've all had these patients with melanoma who look like they're on death's door. And frankly, during my early career, during Chris's early career, this was, metastatic melanoma of the brain was like having a GBM or maybe even worse. I mean, it was just terrible. These patients died very quickly. Well, now you've got these people, you give them radiosurgery and you put them on a checkpoint inhibitor. And I've got patients that have been alive and disease-free for years. So it's a totally different ballgame. And the same is true with some of our non-small cell lung cancer patients, not to the degree of melanoma. But what's different is those tumors do not have an immune suppressive environment to the degree that GBMs do, and they all have tumor infiltrating lymphocytes at baseline. So that's really important, because an immune checkpoint inhibitor can only deregulate the suppression of immune cells if the immune cells are in the tumor. It doesn't actually cause them to migrate towards the tumor. Okay. Justin, do you have a question, or did your just mic go off? I wasn't sure. At any rate, so as a result, we can see that there's differences in the cytotoxic cells that are present. And neutrophils, some degree, macrophages obviously are present in these tumor cells beforehand, and unfortunately they're immunosuppressive. They're not really the kind of macrophages that are helping us fight the tumor. So then with this new information from RNA-seq, we're able to go back and use a software package called David, where you can actually try to figure out, well, what pathways are being upregulated? And again, similar to the results that we got from that earlier microarray data set, which is obviously useful, but a much broader paintbrush, we can see that we're seeing the same kinds of things: the immune response pathways, neutrophils, chemokine-mediated signaling pathways, inflammatory response, antigen processing. These are the pathways that are upregulated after treatment. So it's what we're hoping for. It's what we, as neurosurgeons and neuro-oncologists, want to see. We want to be able to get this immune response in our tumors happening. And then there's another program that actually looks at biological processes that are specific to the immune response. And we were able to do this and look by just a simple Spearman rank order analysis and try to figure out which ones were the most important. And again, similar kinds of findings. Again, immune response, inflammatory response, adaptive immune response, T cell receptor signaling pathways. So again, important things. And T cell chemotaxis speaks a little bit what I was talking about earlier, where you're getting the T cells into the tumor, right? So that they have a chance to do something. When we looked at those genes, over half of them correlated with patient survival. I'm sorry, of the ones that correlated with patient survival, over half of them were immune response pathways and related to different myeloid and particular cytotoxic and T cell populations. And if we looked at the sort of upstream regulatory pathways of those genes, what got those genes to be expressed? All right, they were related to both antiviral response, so that's the sort of type 1 interferons that you may remember from medical school, and then also from the adaptive immune response, so-called type 2 interferons. And so it was pretty cool. It looked like that this interferon upregulation was really related to the positive survival responses. So the story, the picture is beginning to fit together nicely, and that was very heartening. So what do you got to do next? Well, we wanted to, try to understand more about the nature of the immune response, because we've got to be able to, we're not there yet, right? We've got to be able to manipulate this response so we can better fight these tumors. And so different ways you can go about it, right? One was to further engineer the virus to express foreign proteins. Two was to increase the ability of the virus to replicate within the tumor and provoke a better antiviral response. And three was to add additional medications to your treatment to see if those could help augment the impact of the virus. So we were working in the lab back then, and here's some of the people that I had the pleasure of working with. Yancey Gillespie, a well-known PhD neuro-oncologist, Bernard Reutemann, father of HSV molecular virology, Rich Whitley, a famous virologist here, and Wexelbaum already mentioned about. So really one of the things that I have to, again, comment to those of you who have the opportunity to develop these kinds of collaborations, whether in clinical research or laboratory research. These are very rewarding parts of your career, and I encourage you to seek these out. So we developed this virus that did everything that the GTO7 virus did, but it was different in two major ways. First of all, and most importantly, expressed a cytokine. So we actually took advantage of the fact that there was room to put a foreign gene in the virus, and we added interleukin 12, IL-12, human interleukin 12. And the IL-12 is important. It's a cytokine that actually calls in a T cell and an NK cell kind of response, incites the production of interferon gamma. And we also made the virus replicate better. And you can see that on this graph. This is a virus that's kind of like GTO7, and it's replicating in vitro. And here's what it looks like with the virus, the IL-12 virus. So not only is it making IL-12, but you're getting more replication. We had to, as I mentioned back then, the FDA is requiring us to use primates. Thank God we don't have to do that anymore. But you can see that we injected the virus directly into these primate brains that are very sensitive. And sure enough, we got an inflammatory response to the viral injection. And this is sort of what you would expect from just taking and injecting vehicle into the brain. There is a little vacuole here related to it, that process, the stereotactic inoculation. And there's inflammation, which is what we wanted to see, but there's not widespread inflammation. There's not a lot of neuronal dropout, which is the things that we needed to make sure we showed. And recall that when I first saw this, I thought, gosh, I thought there'd be even more inflammation present than there is. But remember, this virus is not being injected into replicating tumor cells. So the virus can't really replicate in the normal brain. So there's not as much viral replication, not as much IL-12, so not as much inflammation. So pretty much what we were hoping. And then we looked at the virus, and this graph is just to remind me to tell you that we looked at a variety of similar viruses that expressed other cytokines like IL-4 and compared them. And the IL-12 virus is this, it clearly was the winner here in terms of how the animals did. So we had the opportunity to do a clinical trial of IL-12, and of the IL-12 virus. And I will tell you, I've learned so much over the course of my career about how to do clinical trials. We had to get the virus produced. I mean, this was years and years of work. And meanwhile, we have a practice, right? We're taking care of patients. We're dictating clinic notes, whatever, and operating and educating the residents and so forth. And meanwhile, oh yeah, and you get back to your computer and you got to say, I got to write another grant to get more virus made. So it's while it's difficult to do both as a neurosurgeon, it is possible, and we've got to be the ones to do it. I just want to say that whether it's neuro-oncologists or neurosurgeons, the people that really understand the disease of glioma. Otherwise, we're going to have others doing it for us, and they're just going to take things and sort of modify them as the way they see fit. And GBM will probably remain the bane of our practices forever. So I want to encourage you to, no matter what your particular area is, to really look at, what opportunities do I have to do research, because you understand that disease entity like no one else, So anyway, we struggled through we slogged through the valley of death here, and were able to get the virus made. And this took years. One day I got a phone call. The NIH was making the virus for us, and they were almost done with it. And they said, Jim, we got to talk to you. And I said, oh, this doesn't sound good. What's going on? And I said, well, got a little bit of bad news. The virus was almost ready, all ready to be vialed and so forth. And then somebody misunderstood the protocol, and they added bleach to all the vials. they killed all the virus that we made. It was like, oh my God. So again, sometime I'm going to write a book about all this, I guess. So anyway, we were able to go on, get the virus made. We developed a protocol for recurrent malignant gliomas, and it was a pretty much a simple infusion design. Um, we set this up like it was convection enhanced delivery. I don't call it CED because there are people out there will say, can you prove it's really convection enhanced delivery as opposed to infusion? And it's not a battle I want to fight. So, but we set it up just like we did for other CED trials that we have done for other agents. So we put 4 catheters in and infused over 6 hours. And we didn't do 72-hour infusions because we had some evidence that if you infuse continuously like that, that the virus that you infuse for the last 24, 48 hours comes into an environment that already has the interferons upregulated and is not very friendly for the virus. It's not a good environment to encourage viral replication. So get it in early and then stop the infusion. And so we treated 21 patients in this study through COVID. It was a mess, again, a slog, but we got it done. And we had some good responses, and we're still doing the analysis right now because we still have some survivors that we're following. But here's an example. This is the very first patient, and she was treated at the lowest dose, and she had progressed, and so she got started on the M032, okay, and then there was a question of whether she had some pseudoprogression, right? So she was given some Avastin to help her through that. And that happened pretty early. It happened about a month afterwards. And so the question is that progression? Is it pseudoprogression? So they put her on Avastin. So they put her back on Avastin. Remember, she'd been on Avastin, failed it, and then the virus was given. Well, I don't know if any of you have rotated through neuro-oncology or whatever, but once Avastin has failed, it's not like you stop it and then you restart it again and you get this miraculous secondary response. So something was different. And for 9 months, she was on the Avastin. And then we took her off the Avastin. 2 months after she was off the Avastin, she still had a pretty good looking response compared to how she had been. Got no more treatment and then survived for 6.5 months. So for the first patient, that was very heartening. And you can also see on these early patients that if you look at what was happening, this is a different patient that we had. We actually did a resection on and looked at the tumor. And what we could see is that this is a quadrant from a flow cytometry study, okay? And you can look at this analysis and see these are central memory T cells, these are naive T cells, these are effector memory T cells, and these are just effector cells. And you can see as you go out from treatment, I'm sorry, these are serum studies, I misspoke. As you go out from treatment, you can see that there's a change in the T cell profiles, and you end up with larger number in particular of central memory cells, but particular T effector cells as well. So interesting, again, sign that maybe something positive is happening. And then, here's some kind of interesting studies that we look at. And this is viral therapy is going to be rough by the RANO criteria and the even the IRANO criteria, because this is the kind of thing that you can see where you see here's the treatment, and then you can see this kind of lysis going on in the center of the tumor. But then there's this inflammatory response occurring around the tumor. You're like, God, is that just tumor progression or what? So, but you can see this big T2 change within it, like there's destruction of the tumor going on. And so you kind of have to either wait this out, or oftentimes we go in and resect those tumors, and we find out, that's when we find out that there's all these inflammatory cells. I'm going to switch gears a little bit. So that virus has gone on and been studied in other model systems. And that's one of the points that I want to come back to in this, is that if you can study something in different model systems and come up with similar results, it adds credence to the fact that you're producing the result that you think you are. And so Renee Chambers pictured here, here she is with some dogs. She was a veterinarian who went back to medical school and did a neurosurgery residency, but she's always maintained her interest in her veterinary aspect of her life. And she actually set up a consortium of vets across the country and developed this phase 1 study of the virus in canine glioma. Here you can see a dog MRI scan, and so these patients, these dog patients, get resected and get the virus inoculated. And she did two arms to this, and the first arm is completed, and she's published this, where she looked at giving the virus just by itself. And she again showed this immune response. They looked at necropsy. And now she's doing it with a checkpoint inhibitor. Turns out there's a checkpoint inhibitor called IDO, which is important in gliomas and in HSV. And so she's giving this particular checkpoint inhibitor with this second cohort of dogs that are being treated, and we'll have to see how that turns out. So we're waiting to see that. But so far, when she looked at necropsies, again, it's her study, but she was able to show that there was an increased inflammatory response present in those cells. All right, now I mentioned this earlier. Kevin Cassidy is again, I collaborated nationwide, and Kevin had a unique idea. He used to be at UAB with me, and he had this idea, well, maybe we could increase viral replication and still keep the virus safe by deleting the viral neurovirulence gene, okay? And so that's what's been done here, but then putting in a different gene from a cousin virus, human cytomegalovirus, CMV, which is a different beta herpes virus than HSV. It's not so neurovirulent, but to see whether in fact that would allow this, this virus to replicate and not produce toxicities. And so, so he designed this virus, and it turned out that it looked like it did well. And I'm not going to spend a lot of time on this, but what you can see is the viruses that don't have this neurovirulence gene, like GTO7, like M032, they don't make any proteins in certain systems, whereas C134 looks like wild type virus and does. So that implies that the virus is going to replicate better. And when he looked at this in various model systems, in fact, the virus did replicate better than the first generation viruses. Here is one tumor, for example, that really showed that effect. Very minimal replication of the first generation viruses, but C134 and the wild type replicated really well. And lo and behold, when you look at mouse survival studies in some certain, those kind of tumors that were not favorable for replication, the C134 did really well. So the key is, well, why doesn't it replicate everywhere? And it turns out that C134 is sensitive to interferons. And so that, and most human glial tumors lack the expression of interferons. So as a result, it allows the virus to replicate in those tumors that don't have interferon expression, but limits its ability to replicate elsewhere. And again, here you can see that here's a wild type virus, and basically the phosphorylated stat is just a measure of interferon expression. And when there's phosphorylated stat present, C134 doesn't replicate. So that's how it maintains its safety. And I'm just going to kind of move on through this right now. So, so we went ahead and started a study with Kevin, a phase 1 study that is currently in progress. We've treated 8 patients right now, and remains to be seen whether this is an approach that we're going to want to use going forward or not. But it certainly looks like it has the ability to increase the ability of the virus to replicate. So, you know, as we approach the end of our hour here, oncolytic viral therapy is here and in the clinic. You know, Imlygic is an HSV that expresses GM-CSF. It's been approved by the FDA for use in melanoma, so it's being used. And in fact, there have been studies that suggest, just as I mentioned earlier in this talk, that using it with a Keytruda or Nivo, these checkpoint inhibitors, may help you have a better response. So, so that's a fact. And then another virus is G47 Delta. This is another virus that was actually invented in Martuse's lab, Dr. Martuse's lab, after I left. And a Japanese neurosurgeon there worked on it, Dr. Toda, and he went back to Japan, and he's just received a conditional approval in Japan. So it's approved. It's just a conditional approval, where he had a remarkable response, and he took advantage of an opportunity there, which I think is really smart. And that is, there's a healthcare system, so they didn't have to pay for all these, but he was able to admit the patients and give them repeated stereotactic inoculations of the virus. So you can imagine, have them come back at a month and then get an MRI scan and say, okay, this part of the tumor is controlled, but this part's not. So I'm going to inject the new part that's growing. Okay. And he did that 6 to 12 times. And these are recurrent patients. And of his recurrent patients, 92% were still alive at 1 year, which is pretty impressive for a recurrent group of patients. Now, obviously, they're pre-selected to some degree, right? But still, very exciting data, and he's just published this in Nature Medicine. And then across town from you all, Nino Kyoko's been working on the RQ-Nestin virus, and it has a lot of similarities to that C134 virus. Again, it takes advantage of using a different promoter to express the viral gene that allows for replication, but minimizes neurovirulence by using a promoter that should only allow the virus to replicate in stem-type cells in glioma. And he also gives cyclophosphamide in the second arm to try to prevent the initial immune response against the virus, which can interfere with virus replication, and then allow a secondary immune response. And then Greg Friedman here at UAB published this paper study that he conducted, and he's a pediatric oncologist, and he was very interested in the problem of pediatric brain tumors. And again, a lot of similarities to those of us who are adult tumor surgeons in terms of these tumors and lack of available therapies for them. And the same kind of thing that I was speaking to you before. These tumors before treatment are cold, there's not immune response present, and then after you treat them, you can make them hot tumors. Interestingly, I didn't know this, but pediatric gliomas are actually colder in terms of antigen presentation availability than adult glioblastoma is. So that was a new education for me. And so this is Jim Johnson, a pediatric neurosurgeon here at UAB, and he did the surgical part of this study, and they treated 12 patients here, and they treated the first 6 patients were treated without radiation, and the last 6 were treated with radiation, and they got the virus infused. And here you can see that they didn't have any terrible toxicities, and it seemed to be safe even at the higher dose that they tested, 10 to the 8th, with 5 gray of radiation. And it seemed like it was promising. If you look at some of the radiographic responses, you see this kind of Swiss cheese appearance occurring over time. Here's a patient 34 months out after treatment with G207, and you can see that this ongoing pockets or bubbles continue to appear even long after treatment. It wasn't, this patient just got 1 dose of the virus. And here's another patient who had pseudoprogression. You can see this radiologist here that worked with him, in fact, came up with a way of looking at the vascular response within the tumor to try to predict pseudoprogression. And you can see here's this patient ended up having resection because of this at 1 month and had some immune cells present. So these are the 5 findings that Dr. Friedman felt were important, that they had increased CD8 cell infiltration, increased, that they didn't necessarily occur at the inoculation sites only, but if you inoculated one part of the tumor, you could actually see it sometimes in other parts of the tumor. So that's key, right? They also sometimes saw evidence of CD4 cells being increased, and it didn't, they weren't always dependent upon having co-stained HSV present. So that's important, right? If the HSV is no longer there or was never there, and you still have the immune response present, that implies that something is happening against the tumor, okay? And then the checkpoint inhibitors were upregulated, again, suggesting maybe that's something to look at. Again, here's just, before treatment, showing some of the same things that I've showed you before. And the adult tumors actually took place in the pediatric tumors as well. You can see that there was increased CD8 cell staining in many of these tumors after treatment when they went on and resected them. And this is a patient that they treated, and you can see where the different catheters were placed in the tumor. And then the patient had a recurrence that was deeper in the corpus callosum. And when they looked at that corpus callosal tumor, they still saw an increase in CD8 cells at the time that was biopsied much later. So, so no virus was placed there, and yet there still seemed to be an immune response that developed. So that's very heartening. If you look at what it looked like before biopsy, you can see there really wasn't this staining present. Here's 4 patients that show that in some cases they saw an increase in CD4 cells as well, and that might turn out, we have some work that's suggesting that CD4 cytotoxic cells might be important as well in the immune response. and in these areas that were far out from treatment, there was an HSV staining present near these areas. So, and in fact, we have scattered HSV staining that's present later on, but it's not, does not seem to be a predominant force. And so I think it's important in just uh, realizing that that there is evidence that maybe this is really an anti-tumor response that's occurring. And then finally, we get an increased expression of checkpoint in proteins. And again, I think that just to see this is pre-treatment and post-treatment. This is a multiplex expression. And you can see if you go through and statistically look at these, you can see that, for example, the checkpoint IDO seemed to be increased, although it wasn't significant in this one patient. And PD-L1 was also increased. So it gives you something to think about that, maybe we can use these viruses with checkpoint inhibitors and accomplish more. Greg concluded that it was safe to look at these, to treat these patients, and there was some exciting survival data that he was able to find. There was evidence that it produced some immunologically important effects, and that in this small group of patients, it seemed like patients who seroconverted from negative to positive might have better outcomes. But again, that's really preliminary. And he didn't find any evidence of viral shedding in his patients. For pediatric patients, they're looking at cerebellar tumors, such as medulloblastoma. And he's in conjunction with the Pediatric Brain Tumor Consortium, they're developing a phase 2 study to give the virus to a larger number of patients to kind of emulate many of the conditions from that New England Journal study. And obviously, there are other strategies that he wants to look at. We have an upcoming study in adults with M032 with pembrolizumab, checkpoint inhibitor. Merck is funding that with us, and so we're excited. We've gotten FDA approval for that and hope to enroll patients in that. And one of the other exciting things about that study is that we have gotten permission from the FDA to transition. Initial patients will be recurrent, but then we'll be able to transition to the upfront setting. And that's really exciting to us when those patients will have a better chance for an immune response. So I think, in conclusion, even in the era of immunotherapy, GBM remains a formidable opponent. Checkpoint inhibitors studies, I didn't mention this, but they haven't really shown much of a response. There's one study using it as a neoadjuvant that suggests that there's a possibility of checkpoint inhibitors alone, but we think that this combination of being able to use the virus to get immune cells into the tumor interstitium and then coming back with checkpoint inhibitor has promise. Which checkpoint inhibitor remains to be seen. But we think that, again, this is an example of how you and your careers can take an idea and really change the physiology of how a particular disease process occurs and hopefully make an impact on it. There's a lot of people who did the work in here, and I want to make sure that I give them due credit. I'm sorry I spoke so long. I realize I only have 2 minutes, but if anybody has any questions at 2 minutes, I'm happy to try to take them on.
Well, Jim, thank you very much. It's a huge amount of information, and obviously you've worked the collaborations really nicely to bring this field forward. And we can entertain a couple of questions. Maybe one quick thing from my standpoint is delivery. These have always been, ever since inception, have been delivered through the catheter in the bed. Has there ever been work done looking at other delivery possible techniques, CSF? Because in vascular, we're having better access for CSF delivery, CSF devices, and also obviously direct vascular, arterial or venous.
Right. No, I think this is a key point, Chris. And so a couple things. So Dr. Kyoko years ago published a paper with a couple of other people at MGH and suggested that it may be difficult to use viral access through vascular delivery because of the presence of the complement system. But that work has sort of been contradicted in part by work done by Yuman Fong, who looked at HSV in colorectal tumors that were metastatic to the liver and showed with vascular delivery that they were able to have an impact on those tumors. So I think that the jury is out on that one. I'm working with Mike Caplet at Cornell, and we're looking at trying to figure out if we can develop a way to deliver this through the blood-brain barrier and see if we could, because I think, the great thing would be to say, come on in, and instead of doing brain surgery on you, I'm going to have Dr. Ogilvy or Dr. Harrigan or someone take you to the lab, the vascular lab, endovascular lab, and just inject this in the carotid. And so I think that those are things that we're looking at, and CSF delivery is also a possibility. We actually have Greg has worked on this for medulloblastoma, and we are actually have, after a long time of trying, to finally developed a good LMD model in a syngeneic mouse of breast cancer that's metastatic LMD. And so I think that these are things that we're anxious to look at in more detail.
Any other questions, Raphael?
Yeah, this is Raphael Vega here. I do a lot of the neurosurgical oncology work here at the BI. So I'm also interested in viruses, and our site here was interested in doing the PV RISPO, right, study with the trying to pre-initiate the immune response, right, with seeing if that will then hopefully stimulate the ability for the viruses to work better. And your work has been very lovely. I've been following it for years, actually. So I guess two things I wanted to ask was, what is your thought of trying, or what are other possibilities of pre-stimulating the immune response to see if you have a better reaction with the virus? And also, what is your thoughts of using LIT to open up the blood-brain barrier, then to use some viruses? I know they're doing that, or trying to do that in Florida, right?
Yeah, so I think LIT is, I'll just take that one first. I think that's very interesting because, of course, there's an inflammatory response produced by LIT alone, right? Virus or no virus. And then, as you say, there is this opening of the blood-brain barrier. And I think that what I see with that is that we'd have to sort of select carefully when we did that, because you don't want to have such an immune response and inflammatory response in combination to the one that can occur with LIT anyway, right? Where you can get some edema associated with it, that it overwhelms the patient's ability to tolerate that much extra volume, if you will. But I think in smaller tumors, that might be a great approach, number one. Number two, I think in terms of pre-stimulating the immune response, it's, again, I think this is the kind of direction that we need to be looking at. We have to figure out, well, how do we do that so that we actually improve the immune response against the tumor? We've looked at rituximab and published some data on that. And if you give it at the wrong time, you actually suppress the response. But if you give it at the right time, you can augment the response. There's lots to learn. And unfortunately, I have one more comment on that, is that mice are not people. And that seems obvious, right? But we all do this, right? We take data from the lab that was generated in mice, and then we act like it's going to be the same in people. And of course, it's not. And a good example for us was one of the reasons we chose to resect those tumors in that middle study at day 2 or day 5 was because in the mice, it looked like the best viral replication was occurring 2 days after treatment. Well, it turns out in humans, our patients at day 5 had better viral replication than ones earlier. So it takes longer for it to ramp up in people. And I think this, again, is something that we need to keep in the front of our brains, that as we plan these studies, that we need to use the data that we generate from mice, but realize also that we humans are distinct and unique.
Yeah, and on that note, there's something I've always wanted to ask, and that is, what about the use of the viral therapy intra-op? You have a recurrence, you're taking out the tumor, and you have the opportunity to inject multiple sites versus doing like a catheter-based CED approach or whatever. Like, I'm sure that's always been something considered, but I don't know.
We actually did that. We did that in that phase 1B study. We resected the tumor. I didn't show the slide, but I've got a little tuberculin syringe, and I went around injecting. I made a grid with half by halves and injected the tumor cavity in the same distance approach with equal volumes at each side around the tumor cavity. Mm-hmm. And it worked. You have to be careful, if you're near the Sylvian fissure. You have to be careful if you're near the ventricle. There are things that you have to think about. But we definitely have done that, and it certainly is something that we're actually going to do that in that checkpoint inhibitor trial. We're going to give multiple doses, but
Yeah, makes sense. Well, thank you again.
no, thank you. Thank you for your questions. Those are great.
Well, Jim, the time is up, but others may be in contact with you if questions come up during the day or a discussion of your lecture. But I really appreciate you taking the time to do this. It was a great update for me, quite honestly. And it was really a huge amount of work you've done. And thanks so much for taking time to share it with us.
Well, I really appreciate it. I know it's highly specialized, and I guess the closing message again is that we have the best laboratories in the world, and we should all take advantage of the fact that we have the opportunity to learn more about the human brain and our diseases. We have a special responsibility, I think. So, thank you.
Well, thanks very much, and I hope I see you in Colorado.
Yes.
We'll see you in a few weeks. Thanks.
That's right.
Thanks very much.
Thank you.